So with this video, we're going to turn from talking about the organs of the lymphatic system and the flow of lymph through the lymphatic system to talking about immunity, how we go about defending ourselves from attack. And before we get started, we've got to get a couple of definitions out there. So a pathogen is a disease-causing agent that can threaten the body. And so typical pathogens include bacteria or fungi, so yeast infections or protozoans or viral pathogens. So anything that can cause disease is a pathogen. That term antigen, we were introduced to it back in the chapter on the blood. Antigens are identifier molecules that are found on the membranes of the surface of cells. They can be proteins or polysaccharides or a combination polysaccharide protein. That's what glyco means sugar. It was about sugar. Protein is protein. Or glyco, and I'm guessing you can tell me what the second part of that is. So that would be a saccharide or a polysaccharide and a fat. And during prenatal development, so early in the developmental process in the uterus, a baby's body learns what antigens are self-antigens, indicating that the cell and the antigens that are found on that cell would be a part of the body. And this allows then the immune system later on, once the baby is born and it begins encountering pathogens, allows the baby's immune system to compare to what's already been logged in as a self-protein or a self-antigen, and then will attack anything that is non-self. And so then that lets us talk now about two kinds of defenses in the body. First are the innate defenses, often talked about as the non-specific defenses. These are the defenses that are present at birth, so that's what we mean by innate. And they don't distinguish among threats to the body, so they're not tuned to go after a single specific threat. Instead, they work against general threats, threats of a lot of different varieties. The second kind of defense that we have in the body are what are referred to as the adaptive defenses. These are defenses that learn to recognize a particular threat. That's why they're adaptive. They can learn. And we also talk about them as the specific defenses because they go after particular threats. So, for instance, if you get a vaccine for the measles, then you are training your body to recognize that particular virus and be able to defend against it. That's what we mean by that kind of specificity. Adaptive defenses are provided by lymphocytes. So we'll have to be talking about those lymphocytes in the next video, where we start looking at the adaptive defenses. And it's the lymphocytes that are trained to target very specific pathogens and to ignore any others. So we have to have a lymphocyte that's tuned specifically for that an antigen. But if it's tuned for an antigen, it won't be sensitive to any other antigens. And most of these adaptive defenses, they develop after birth and following exposure to some sort of non-self antigen, so bacterial invasion or viral invasion of the body. Lead to, sorry about that, goodness, will lead to adaptive defenses. Yeah, I almost dropped my my laptop or the notebook here. Okay, so let's now get into some of the innate defenses. And the first one is what we talk about as species resistance. Humans have species resistance because we are not affected by bacterial or viral pathogens that go after other species. For instance, feline leukemia. It's a virulent disease in cats. It'll kill the cat, but it's not passed from cats to humans. So we have species resistance against feline leukemia. So in order for a virus or a bacterium to affect a human cell, it first has to bind to that cell. And if the pathogen doesn't have receptors or binding sites for the antigens that are found on a human cell, then it can't damage a cell. It can't latch onto it and do its damage. And so that's what species resistance is. We are protected from many diseases that other organisms get that we will never be able to get. Okay, so the first line of defense then, if we're talking about a pathogen that can affect a human, are the mechanical barriers. And their job is to keep pathogenic organisms and other dangerous materials, toxins, or chemicals that are dangerous out of the body. So if we think of the body as a castle, then the mechanical barriers are the castle wall and the moat that are around that castle. So they involve the skin. There's a barrier that covers the external surfaces of the body. The mucous membranes that line the respiratory, digestive, urinary, and reproductive tracts. So all those openings that open out to the external environment, they could be a surface across which a pathogen might try to gain entry into the body. And so we have those membranes. They're there. They frequently have multi-layers of cells so that we prevent any or make it harder for a pathogen to get into the body. Frequently there's hair or cilia on these surfaces that can help to trap infectious agents and particles. And we may produce sweat if it's skin, or mucus fluids like mucus if it's the mucous membranes. And there's the water in the moat when we think about the sweat or the mucus fluids that, or maybe the tears of the eye that help to wash away pathogens from the body surface. So our first line of defense are these mechanical barriers that help to prevent anything from gaining access. And so if we have a scratch in our skin or a tear, say, in the vaginal lining that occurs during sex, then that provides a point of entry then for whatever pathogens might be out in the environment to get across that skin layer or to make it into the deeper tissues wherever that damage occurred. So let's say something gets into the body, then the second line defenses come into play. And these, again, if the human body castle, these would be the guards inside the human body, or inside the castle, that will notice, hey, you don't look like you belong, let's take you out. And these second line defenses include chemical barriers, a type of white blood cell called a natural killer cell, the processes of inflammation, the work of macrophages and neutrophils in phagocytosis, and, last of all, the effects of fever that act as a defense mechanism as well. So we're going to talk about these 5 different second-line defenses that we find in the body on the next 5 slides. So we'll start first with chemical barriers. We have enzymes. Enzymes are chemicals that can help or that break chemical bonds. And so we find in gastric juices and other digestive juices. And in our tears, we have enzymes that can help break up any bacteria or viruses or other materials that try to get into the body. In tears, this specific enzyme is called lysozyme. And then in our digestive juices, we have a number of enzymes that are there that help to break down food, but in the process can also break down bacteria or viruses as well. We use low pH as a second chemical means of preventing dangerous materials from entering the body. So when we swallow food, maybe there's some bacteria that are on the veggies from the veggie bar or from the salad bar at a restaurant. There's a good chance that the pH of the stomach will also help to kill whatever the pathogen is, because low pH denatures proteins, and when you denature a protein, you take away its function, and so if the bacterial proteins on the surface of the bacteria or inside the bacteria are damaged, then the bacteria is likely to die. Interferons are a third kind of chemical barrier. These are chemicals that are produced by a cell when it is invaded by a virus. And the interferons leak out into the interstitial fluid around the cell, and they are a cue to other nearby cells to initiate processes that block virus replication. So if one of those cells, one of the neighboring cells, happens to be invaded by a virus, then it would be harder for the virus to take over the machinery to begin making all the components of the virus, which is what happens inside a cell that's been infected by a virus. So interferons are chemicals that prevent viral replication. Sorry about that. And by releasing this hormone-like chemical, a virus-infected cell can kind of provide an early warning to neighboring cells that are most likely to be infected by that virus next. Defensins, these are chemicals made by white blood cells in response to locating a non-self antigen. And so a white blood cell that notices, say, a bacterial cell will secrete a bunch of defensins, and those are enzymes that can help make holes in cell membranes. So it helps to punch a hole in the bacterial membrane and kill the bacteria. Collectins are a fifth kind of chemical barrier. Collectins are released, and then in response to non-self antigens that are found on pathogenic materials, and they bind to those non-self antigens, and they make it easier for white blood cells, for neutrophils or macrophages to come along and ingest that whatever the particle is that's bearing that non-self antigen. So white blood cells will more easily phagocytize bacteria or yeasts or viruses if they have collectins bound to them. And then last of all, among the chemical barriers are a set of chemicals called complement. And these are a group of proteins that are plasma proteins, and they're found in blood plasma, and they also make it out into interstitial fluids. And along with antibodies, they help to stimulate inflammation, they attract phagocytes, and they even enhance phagocytosis once the phagocytes are there. And it turns out that much of specific immunity is really enhanced and performed thanks to the complement proteins that are found in the blood or in the tissue fluids. So there's the term, if something complements something else, then they're great partners, they work well together, they match each other. And so we think about complement as being complementary to the adaptive cells, the adaptive immune response. Okay, so chemical barriers were one barrier, one of the second-line defenses. Natural killer cells are a second kind of second-line defense, also called NK cells. So frequently, when people talk about them, they just, rather than calling them natural killer cells, they describe them as NK cells. And these are specialized lymphocytes that will notice if a cell puts out onto its cell membrane surface antigens that alert the body to either the fact that the cell has become cancerous or that a virus has infected that cell. And so the natural killer cells recognize a defective cell, a cancerous or virus-infected cell, by those identifier proteins, and when an NK cell encounters one of these defective cells, they will secrete, again, enzymes called perforins, which are molecules that can poke holes in the cell membrane and lyse the cell, break the cell up. And it's only the cancerous cells that escape detection by NK cells that are able to grow and multiply and even metastasize or spread from one location to another. So most cancers are recognized by NK cells and taken care of. So it's a good bet that all of us have had cells that have become cancerous, but our NK cells have taken care of it for us if we have never expressed any cancerous tumors in the body. Okay, so we have chemical barriers, natural killer cells. Third of the second line defenses is inflammation. And inflammation is caused when mast cells or other lymphocytes release two kinds of chemicals, a histamine and another chemical called heparin. And we think about four signs and symptoms of inflammation. One is swelling. That's because the histamine makes the capillaries nearby, wherever the mast cells or whatever cell is releasing histamine, that area where the cells are, well, the capillaries there will become extra leaky and more fluid will get out than is called back in, and so you get localized edema. It also increases blood flow to the area and prevents the heparin helps to prevent clotting from happening. But that increased blood flow brings extra heat, and with the capillary networks really engorged with blood, then the region, if it's at the skin surface, we can see redness. And then last of all, the fourth, and this is the symptom, the edema, redness, and heat are all signs that can be recognized by a caregiver, but pain is a symptom. It's only recognized by the patient. And pain is caused because all that swelling makes for pressure, and if there are damaged tissues nearby to where all this is going down, those damaged tissues will release a kind of chemical called a bradykinin. And these bradykinins help to stimulate pain receptors, and that leads to the pain that we associate with inflammation. So the whole, all these conditions of inflammation speed up the activity of phagocytes and bring in more phagocytes into the area because we get a lot of blood flow into the area. And as the phagocytes in that area encounter and engulf and destroy pathogens, then if you remember neutrophils, they could phagocytize somewhere between 12 and 24, 1 to 2 dozen bacterial cells, and at that point they run out of supplies to destroy their prey, and so they become essentially useless to the body. As those white blood cells die, then we can see accumulations of fluids with lots of dead white blood cells in that area, and that's what pus is this accumulation of fluids with dead white blood cells. So all of this inflammation creates congestion in the area. Fluid flows in, but it takes a longer time for fluid to get out. That helps to quarantine the area, gives the macrophages that are called in, the neutrophils that are called in, a chance to go roam around and find any pathogens and remove them. But prolonged inflammation can lead to the development of a network of fibrin molecules or fibers. Remember, fibrin is one of the part of the production of fibrin is part of the processes of coagulation. So that's that network of fine protein fibers, and so we can get a fibrin network of like the beginnings of a tissue to begin to develop around the affected area, and then fibroblasts will move in, and they can start producing connective tissues among the fibrin fibers, and so eventually a little cyst can form with a connective tissue capsule around it. Okay, so that's inflammation. Let's move on to the next of the secondary defenses or second-line defenses, and that's phagocytosis. So remember, it's white blood cells that are responsible for phagocytosis, and there are two kinds of white blood cells that phagocytize, and they will engulf damaged or abnormal body cells and any foreign cells that they encounter or any materials that don't belong in the body. So one kind of neutrophil, one kind of white blood cells is a neutrophil. These are the smaller ones that circulate in the blood and engulf really small particles or bacteria. And then there are the monocytes that circulate in the blood, and then after 24 hours of that, they will do diapedesis. They will make their way into the tissue fluids, maybe take up residence in specific organs, and their job out there is to engulf the really big stuff. At which point we talk about them as macrophages, and almost all of our tissues have residential populations, fixed populations of macrophages, the ones that never leave, and then we have the visitors, the ones that are free to travel. And so most organs and tissues have a little bit of, some of each kind that are hanging around and making sure that nothing pathogenic survives for long. Okay, so the last of these second-line defenses is fever. Fever is the sustained elevation of body temperature above whatever an individual's normal body temperature is. So if we think of normal body temperature as being 98.6 degrees F or 37 degrees Celsius, then it's anything higher than those kinds of numbers. Now, fevers are brought on by a class of chemicals that are referred to as pyrogens. Gens means generating or making. Pyro, fire. Hopefully pyromaniac. I also can use pyrotechnic displays. So these pyrogens are chemicals that interact with the hypothalamus and change the set point for homeostasis around body temperature. And it turns out that more and more physicians and other healthcare providers are really suggesting to families, parents, that rather than suppressing low-grade fever, allowing a low-grade, a moderate fever to continue without trying to suppress it can be valuable to helping the body overcome, to defeat whatever the infection is. And there are two reasons that this works. First is that you make the body less hospitable to pathogens. Pathogens have a preferred temperature for the fastest replication and growth, and if you can bump them up outside that preferred temperature, then you can make the body a less desirable place to live and can slow down their reproduction. Fever also causes the liver to take up any iron that's free in the blood. And frequently, iron is a limiting growth factor for bacteria and fungi. And so with less iron, this sequestering is the term that's used, this hoarding of iron in the liver, then makes it harder for pathogens to reproduce efficiently. So by either pushing body temperature outside of the normal preferred range for pathogen, or by removing from the body fluids some needed material like iron, then we can help discourage bacterial or fungal growth and reproduction.